So now we move up a level to the tissues, not as in Kleenex, but as in when we have multiple cells working together, often attached to one another, that are performing together in terms of a function. All right, so like we talked about different cells having different functions, we elevate that to another level as well when we have tissues. These tissues that are helping to maintain homeostasis, right? That stable internal condition. So when we study histology, what we're studying are tissues. So often when we're doing this in the microscope, right, we're not looking necessarily at individual cells, we're looking at an organ, we're looking at tissues, and so for histology, we're looking at those tissues, not usually individual cells. And with these tissues, there's four basic tissue types. We've talked about it before. Epithelial, our covering and lining. Connective, for protecting and surrounding and functionally connecting things. Muscle, for movement. And then nervous, for communication and coordination. So you can see there's a number of different organ systems and organs that have these different tissues in them. Now remember that by definition, an organ is more than one tissue type. So even when we say the brain is basically going to be nervous tissue, yes, for sure, but there's also going to be connective tissue. So these organs are just examples of where we have a lot of these particular tissue types. All right, so in this chapter, what we're doing is we're going through the four basic tissue types, and then we're going to find that there are specific tissue types in those four general categories. And we're going to go epithelial, connective, and then muscle, and then nervous. And you'll be happy to know that they get easier and fewer specific types through time. So we're going to start with the biggest group, which is the epithelials, and then we're going to get into connective tissue and work our way down from there. So we said epithelial tissue is a covering and lining, and so we're going to find that it is often going to be on the surface of organs, either on the inside or on the outside. So covering and lining, so lining the digestive tract, lining the respiratory tract, lining our body. Those are epithelial tissues. And there's two main forms of epithelial tissues. Those covering and linings that we've been talking about before, and then what we also find are glandular epithelia. So kind of like when we were looking at the plasma membrane of an individual cell, right? That's kind of like the epithelial of a cell. And we know that with vesicles, you can have the plasma membrane and then it kind of invaginates and forms a pocket, right? That also happens in terms of our epithelia, and so that's kind of where we get glands like sweat glands and oil glands and mammary glands and things like that. So these are secretory tissue that are made from modified epithelia. So when we look at epithelium, it's covering and lining, protection, absorption and secretion, so things going in or out of an organ, in or out of the body. Sometimes filtration, and then also sensory reception on the edge of an organ or on the edge of us. We want to know what's going on in the surrounding environment, so a lot of our receptors, our sensors, are going to be located there. Okay, so for epithelial tissue, 5 different distinguishing characteristics. Remember, we look at the structure and the function, and with these 4 main categories and even with the specific subcategories, we're going to see those differences. We're going to see the differences in structure, and that's going to give rise to different functions. And epithelial cells, to be sure, are very unique in terms of their structure. So they have what's called polarity, meaning that they're not all the same like our generic cell. They have poles to them, like, you know, the Earth has a North Pole and a South Pole, right? So polarity, they're going to have ends to them that are slightly different. They have specialized contacts, because when we see epithelial tissue, when you'll see it in the microscope, epithelial tissue, there's not a lot of extracellular space. These cells are packed next to one another and attached to each other, often to form, like we said, like a protective barrier, etc. We don't want things slipping by. So a lot of specialized contacts between these cells. They're usually supported and held onto the body or held onto the organ by connective tissues, and they tend to be avascular. So remember when we were talking before, we said that A means without, vascular means blood vessels. So they don't have blood vessels going into them. So they rely on blood vessels right underneath them, and then nutrients and oxygen and water will diffuse from those blood vessels. So, but we don't have blood vessels going into the epithelial tissue itself, but they are innervated. Like we said, we need to be able to sense right at the very edge of things, so we do have nerves and receptors going to it. And then lastly, epithelial tissue is very good at regenerating, because if we're going to protect and it's going to get scraped off and damaged and things like that, we want to be able to replace it, right? We don't want to wear through our skin or wear through those linings at the edge of our organs or inside of our organs. So this figure here shows us some of those specialized characteristics, like we said. So we can see these are called columnar cells. You can see they have a unique shape, but the top of the cell is called the apical or free surface, right? So this might be where the food is up here floating along or something like that. So this surface that's not attached to anything, that's what we mean in terms of free, is called the apical surface, and there's only one. The lateral surfaces on the side are attached to other cells, and the basal surface is like the basement down at the bottom of the cell. So that's what we refer to in terms of polarity. Here we see a basement membrane, some connective tissue underneath that's holding this epithelial tissue on. So there's a lot of interesting aspects about these cells. We say that they have specialized junctions as well. We talked about some of these junctions before. Tight junctions where they're sewn together. You mentioned like that, right? Like seams on a shirt. There's also, at times, gap junctions, which actually allow substances like ions to move. Desmosomes and hemidesmosomes, like half of ones, that occur like buttons where they're attached in one particular place, but you have some space in between. And then there's also these adherens junctions, which are kind of like if you had a big massive button or something like that, it connects it on a larger level. Okay, so now we get down to the subcategories of epithelia. And kind of like, you know, the binomial system for organisms where there's genus and species, and that describes, or even you, like a first name and a last name describes you specifically, as opposed to Churosky overall and different other people in my family that have the Churosky name. Two names that give it a specific type, okay? The first name indicates the number of cell layers. So if it's one, we call it simple, just one cell layer thick, very thin. And if it has more than one, we call it stratified. So think about the strata along the walls of the Grand Canyon, right? You can see those layers of rock or strata. Now, how many strata we have is different depending on the function of the tissue. All right, so first name, number of cell layers, simple or stratified. There's always exceptions to the rule there in terms of that, but we'll see that coming up. Second part of the name describes the shape of the individual cells that contribute to that tissue. So we saw some tall cells before. Those are called columnar. Flattened cells are called squamous cells. So squamous cells, if you view them from the top, they kind of look like an egg over easy or something like that. Egg sunny side up. But much like if you had a plate of eggs and you were looking down at it, it would look like that. And then if you took it and you looked from the side, it would look quite different, right? So it would look more like this from the side. So squamous cells are thin, flattened cells, and it depends on which way you're viewing them as to how they appear, but they're thin, flattened cells. We also have cuboidal cells, as you can imagine, right? They're about as high as they are wide. Now, they don't have right angles like that, but they're about as high as they are wide. And then we have columnar cells, like we mentioned already, right? Columnar cells are going to be higher than they are wide, right? So they look like little columns. Okay. So if we put those two names together, then we can describe the different types of specific epithelia. Simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, etc. Right? So we can see the difference between simple and stratified, right? And you notice that the deeper layers of stratified tissues, they don't have an apical surface, right? Because for the deeper layers, they are touching all other cells. So it's only those that are at the very top that actually have a free or apical surface like that. Cell shape, squamous, cuboidal, columnar. All right, so let's tour through. So, simple squamous epithelium. Simple squamous epithelium, single cell layer thick, flattened cells. So, these are coverings and linings that are as thin as you can possibly get in the body. And if you have a very thin layer, it's not going to be good for protection. You can burst through it very easily, but very good for allowing things to transport through. So, we're looking here in the lungs, and each of these individual cells, here's one cell, a little pancake cell. Here's another pancake cell right there. There's another little pancake cell right there, and there. Right? Each one of those cells is a simple squamous epithelial cell. And so this whole structure, like we see here, this bubble-like structure over here, is called an alveolus. Alveolus. We'll see that when we get to the respiratory system. But we have to have gases exchange here. We have to have gases exchange with the capillary surrounding it. And so we want to facilitate that. We want to make sure that we can have oxygen and carbon dioxide moving very easily across it. So one single layer of cells flattened makes that happen. There are other examples as well in terms of where you can find simple squamous epithelium, but this is a good example in the lung. Simple cuboidal. Now this is a weird one. When you look at it, you're like, simple, it should be one layer of cells. Now remember, epithelial tissues, when we're looking at them in the microscope, they're going to be on the edge of an organ, right? We're usually seeing a slice through an organ or something like that. We're going to look to the edge. So there's going to be a bunch of open space. Then the very first like dark purple or pink layer is going to be your epithelium, right? And so when we're talking about this one, you're like, ugh, I don't see any edge there, right? But these are simple cuboidal cells that are lining tubules inside of the kidney. And so this is a tubule right here. And this is one cuboidal cell. This is another cuboidal cell. This is another cuboidal cell. There's one here and we can't see the nucleus. And one here, we can't see the nucleus. And then one here. So when we're looking at that particular one tubule, right, they are forming a covering and lining. It's one cell layer thick. The urine is in here, or the filtrate, and it's going to be absorbed or secreted across this wall. But this is only one cell layer thick in terms of this tubule. One cell layer thick in terms of the wall of this tubule, that tubule, etc. You're looking at a cross section, so you're looking down the barrel of those. That's why they look like little donuts like that. So simple cuboidal epithelium, one cell layer thick, about as high as they are wide. Simple columnar. Now this is a little bit easier to see. Tall cells, one cell layer thick. Here's your basement membrane down here, right? So this is a little bit thicker. Oftentimes, like in the digestive system, along the digestive tract, it's a good place. So things are going to be able to absorb across. Nutrients can absorb across this. But it's a little bit thicker, so it gives you a little bit more protection maybe compared to simple squamous or something. And this photo is a little bit difficult because here is your open space. So here might be where your food's going to go. And then this is your single layer here. So here's the basement membrane along here. So here's one cuboidal cell. Here's another one here. There's another one there. There's another one there. Okay? So this diagram's a little bit easier to see. This one, you just have to think this is your space right here. So we have one cell layer between here and then where the basement layer is. Okay, this one, we said there's always exceptions to the rule, right? If we look at the title of this, columnar, so these are taller cells, pseudo-stratified. So pseudo means false. So this one looks like it's more than one cell layer, but in fact it's just one. So if we look at the diagram here, so see how there's almost like multiple layers? Here's the nuclei, right? This is a nucleus, this is a nucleus. And it almost looks like, okay, there's a layer of nuclei here, there's a layer here, and there's a layer here. So maybe this is 3 cell layers thick or something. But in fact, each one of these cells, like this cell here, has a little extension that goes all the way down to the basement membrane and then comes up to the top. And this one goes down here and touches at the bottom. This one has an extension that goes to the top and to the bottom. They're irregularly shaped cells, but each one reaches down and touches that basement membrane or that basal lamina. So that means it is a single layer, but it looks like it's more than one. So we call it pseudo-stratified. These ones are often ciliated as well. There are the cilia there. So this is that mucus escalator. So we have mucus up here that's being transported along these cilia, up out of the trachea, etc. And these right here, these white ones here, those are actually goblet cells. They're individual little secretory cells instead that are secreting mucus. Okay, stratified squamous epithelium. This is like in your skin, inside of your mouth, any place where we have a tube that's opening to the outside, we need more layers of protection for those hard Doritos that you're eating or whatever, right? So if we were to count the cell layers, here's the bottom of this epithelial tissue, here's the top, count the nuclei. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 to 30 nuclei that we can see stacked on top of one another. So this is truly a stratified epithelium. We can see down here, right, this is where the connective tissue is holding it on. This is where the blood vessels are. These are much healthier cells down here. In fact, as you get towards the top here, see how it gets flattened? So we call it stratified squamous because most of the cells are flattened. But as they get further away from the nutrients, they start to die off as well. So we get even kind of dead layers of cells out here. Transitional. Now this is a stratified, right? We can see 1, 2, 3, 4, 5, 6, 7 maybe layers if we count up the nuclei all stacked up here, right? So it is a stratified type of epithelium, but this transitional epithelium, if you're going through a transition, you're going through changes, right? And so this epithelium, which is a covering and lining like along the bladder and the ureters, where when your bladder's full, it's going to stretch, and when it's empty, it's going to get smaller, like a balloon, right? If you blow up a balloon, the dark color when it's empty, and then the lighter color when it's full of air, shows you that the lining of the balloon is actually getting thinner. And that's the same thing that happens with this transitional epithelium. So when the bladder's full, it might only be 2 to 3 cell layers thick, stretched very thin. And when it is empty, then these cell layers are going to pile up again, and we're going to have maybe 7 or 8 cell layers thick instead. So transitional epithelium. So when we think about our different types of stratified epithelium, we have pseudostratified. That's a single layer, but it looks like 2 to 3 cell layers. Then we have transitional, which is more like 5 to 10 cell layers. And then we have truly stratified epithelium, which is more like 20 to 30 cell layers. So that's going to help you when you're seeing them in the microscope. Now there are some other ones. These are the main epithelial types. There are some other ones. There's stratified cuboidal and stratified columnar and stuff like that. Those will only occur in very unique spots of the body, so we won't worry about those really. They do exist though. Okay, lastly, besides covering and lining, we said that epithelial tissues also help to make up glands, and glands secrete things like mucus, like saliva, like sweat, etc. So when we look at the glands, those can be classified by what they put out, like we mentioned, and kind of where the type of secretion that they're putting, how they secrete it out, etc. So if we look at it, the site of product release as well is going to be important. So endocrine versus exocrine. Endocrine, they basically secrete a hormone. They create a hormone and then it's released into the bloodstream. And then exocrine, it actually goes into a duct or a pipe that takes it into the local area like your skin, or saliva glands that secrete it into your mouth. So long-term transport using a blood vessel in endocrine, and exocrine uses a duct instead and delivers it to the local area. Also, these glands can be classified based on the number of cells creating the gland, whether it's just one, like the one we saw before, like the goblet cells shaped like a goblet, or those that are multicellular instead. So endocrine glands, like I talked about, are ductless glands, not ductless, like quack quack. Ductless glands that secrete hormones, that's their secretion into the bloodstream for traveling long distance and signaling distant cells. Whereas exocrine glands, like we say, do have a duct and they secrete into the local area. And so these are most of the different types of glands that you would normally think of. So the unicellular ones, like we said, are often called either mucus cells or goblet cells, and they produce mucus or mucin, which then combines with water to give you your slimy coating, like in your nose and in your mouth and along your digestive tract, someplace like that, urinary tracts. And you can see it here. So this is one cell, right? So we see other columnar cells adjacent to it, but this is one mucus cell. And it's going to produce a secretion that then is going to come up here like that. When we have multicellular exocrine glands, which is like sweat glands and salivary glands and stuff like that, they have that duct that comes out of them. And so they can be classified by structure and mode of secretion as well. So the structure, if they're simple, they just have a single duct coming up. Let me show you. So simple, we can see just a single duct coming up like this, as opposed to compound. Compound, we have groups of secretory cells that are secreting into multiple ducts that then come out to the surface. So this is called compound instead, duct branches. And then it's a question of whether it's like tubes, whether it's actually like round, like alveoli, like we see here. Those are some of the ways that our different glands can be categorized. But don't worry, we're not going to have you kind of say, okay, this is a sweat gland. Is it a compound tubular or compound alveolar or whatever? It's just showing you the diversity of these different types of glands. Now, in terms of mode of secretion as well, there's different ways that they can put out their secretion. Merocrine glands just release it from the cells. They go into the duct, and then it comes out the duct itself, like secretory vesicles like we learned about before. In holocrine glands, these ones, the actual cells break apart, so pieces of the cell go into the secretion itself. So you would imagine that the secretion here is going to be much thicker. So like the difference between, there's two different types of sweat glands we'll talk about with the skin. There are eccrine sweat glands, which produce the normal salty sweat, which are all over the body, and then there's the apocrine sweat glands, the stinky sweat glands, and they have a lot of proteins and cellular fragments that are in the secretions, and bacteria eat those, and then that's actually what produces the stink associated with the stinky sweat. So that's some of the differences between, again, modes of secretion, which also allows us to categorize our exocrine glands. So that takes us through 1 of 4, epithelium, but we said that was the most detailed. So create flashcards or whatever you got to do to be able to categorize the name of the epithelial tissue, where you can find it, and its function. Is it there for transport? Is it there for protection, secretion, etc.? So we have the other 3 remaining: connective and muscular and nervous, and we'll do that in our next part instead.